Thank you for joining us today for our podcast. Our guest today is Ross Young, CEO and founder of Biosorcia Inc. I just want to confirm the title of the discussion with you. It's discussing AI, microbiomes, and 8 orders of magnitude improvement. So I want to welcome Ross to the WATSNEX podcast. Ross, you're a serial inventor and true pioneer. For our audience, can you briefly introduce yourself and more importantly explain the monumental problem that Biosorcia was founded to solve?
Terrific. So again, Ross Youngs, the company's Biosorcia, and my background's environmental science and industrial engineering, worked in the optical and video disc fields. I've founded a couple different companies. So I've been an entrepreneur since 1988, hold a number of patents worldwide on technologies and products. And ultimately, the problem that we're solving is understanding nature's deep cellular communication in a way nobody's ever been able to understand it. Is that leads to training sets and data around how to design drug-like molecules and how to find drug-like molecules, again, in a way nobody thought possible.
You mentioned that typical research only access 1% of culturable, leaving 90% of the deep dark chemistry hidden. Can you explain what this deep dark chemistry is and why it's so valuable for immune oncology and other therapeutics?
Actually, with ease. So what we would point out is that the Earth, through its entire history of life, has been a microbial world. It was the microbes that inhabited this Earth early. It was the microbes that evolved and ultimately terraformed Earth into the environments we have today, turning a hydrocarbon atmosphere and inhospitable planet into a breathable planet that we have today. It was the microbes that evolved into all life that exists. And it's the microbes that still dominate all life today. It wasn't until 15, 20 years ago that we really started to understand that humans had a microbiome that outnumbered its human cells. When you talk about its microbial cells of 40 trillion to 30 trillion human cells, and that when you talk about the genomics, outcalculated the human. And what do I mean by that? We have 23,000 genes as a human. Our microbiome probably has more than 25 million genes. And even today on this planet, that connection to the microbial evolution, the small molecules that it makes, the cellular signaling that it makes, is dominant throughout the world today, meaning that if you don't count woody biomass, microbes outweigh all other life combined. Fish, animals, insects, birds, microbes are dominant on this planet. And I don't think there's a person alive that knows about science that would claim otherwise, that microbes will probably outlive all other life forms on this planet into the future as well. So there is a deep hidden understanding that is void of our understanding as humans. We've learned less than 1% about the cellular signaling power of microbes to date. That is a black spot we can fill and ultimately not only understand the molecules and the therapeutic opportunities or potentially diagnostic opportunities, but we also could design drugs much better with that understanding we don't possess today.
Wow, thank you very much for sharing all those insights. For section, I would like to do a deep dive in the 8 orders of magnitude leap. I'd like to explore the engineering and business strategy behind the scale and efficiency of Biosorcia technology. Does that make sense?
What I would suggest, and you can't hardly hear, or might get it focused somewhere here. This 100 milliliters of, in this case, it's green tea. Let's call it biological material. The reality of it is that microbes, that's a typical starting point for drug discovery, especially when you talk about culturing. And ultimately, the vast majority of microbes can't be cultured. So even if they are cultured, the problem becomes they won't produce the metabolic pathways that it's been encoded with because it's not in a culturing situation a reason for it to produce a lot of those metabolic pathways. So even culturing doesn't really lead to a whole lot, but culturing has led to a lot of drugs on the shelf. So when we think about this, the small molecules of microbes, which only 36,500 have been documented on the Natural Products Atlas list, led directly, indirectly, or inspirationally to 50+% of the drugs on the shelf. Now, when we look at training synthetic approaches, we're certainly training them on the small molecules of microbes, as well as other training data sets. So the question comes up, is culturing the only way to get at microbes? And the answer is no. We've been able to use metabolomics, genomics to predict. But even with that, the predictions are not that good. We don't really close off all metabolic pathways to a molecule that's defined without having knowledge of what that molecule is. That helps a lot. So there is about 99% plus of the small molecules by microbes we still have no understanding of. So the question comes up, how could you get it? If you can't get it through culturing, if you can't get it through genomics, can you get it through synthesis? Well, we tried that. It was called combinatorial biology that spent billions annually trying to randomly discover molecules that might hit drug targets and ultimately find a next drug. Out of that decades-long experience and billions of dollars, one drug got approved. The reality of it is synthetically random, you cannot really stumble upon it. And you can't stumble upon it because nature has a finite set of language around molecules. It's not an infinite set, it's a finite set. Just like language, if I said to the audience today, I'm going to teach you 2 words in Egyptian, and you are now going to be able to speak Egyptian, nobody would be able to speak Egyptian. It takes understanding of the language in order to speak Egyptian. It takes understanding of the natural priors to be able to speak the language of cellular communication. So that's what we would say is there's a finite set of molecules within nature that can be trained. We know very little of that finite set. Sooner or later, we may know a lot more, but right now we know very little. And that's important when it comes to understanding things. So what can we do? We believe it's industrial scale microbiome mining. So instead of starting with 100 milliliters, one needs to go to the natural environment where microbiomes exist at scale. Now, the problem is they exist at scale in very high dilution. So you may have 0.02 grams per liter of biomass or 0.05 grams per liter of biomass in an aquatic habitat. It may even be lower in a terrestrial environment or on a plant. And the reality of it is you certainly can't capture it from a human. It's not in enough quantity. So we go to the aquatic world primarily, or to a microbiome that can be put into a liquid carrier of some kind, and ultimately collect the microbes. Now, what's the problem with collecting microbes like that? The problem is lysing of cells and speed of collection. If you lyse the cells, you create sludge. If you dwell in your time frame to preservation, you create sludge because all microbes change their metabolic pathways rapidly. In as little as 15 minutes, their metabolic pathways could change. They're trying to survive. So you're looking at sludge. So wastewater and water processing and typical technologies to get at sludge will give you sludge. And it is not very useful. But if you can preserve the molecules and not lyse the molecules at a massive scale, then you're starting to get a complex mixture of microbes, typically between 400 and 800 species, with a dominant species and those that live within the microbiome that ultimately come out of a given habitat's microbiome. But that's a complex mixture with a lot of complexity. So you must have a lot of it. So Biosortia typically starts with a minimum of 8 Olympic-sized swimming pools of a habitat's microbiome, processing it without lysing its cells and preserving it within 2 minutes and 14 seconds in order to have the starting material. Well, now you have wet weight, a lot of material, or if you concentrate it as you process it, which is what we do, you still have a dry weight and wet weight combination, which is a lot of material. So ultimately, you need to dewater it or lyophilize it, which is a lot of material to lye off. When you do, you crack the cells, but you remove the water and it's already stabilized. So now you can get at the molecules that are there. But what do you do with a massive amount of molecules in the biomass? Well, you've got to process it. So industrial scale microbiome mining isn't only about getting the microbes, but it's about processing the microbes so that you can get at the small molecules, move the proteins this way, move the fats this way, but get the small molecules through various extractions and fractionations. Ultimately, that allows you to do analytics on those molecules, discover what is known, what is unknown, but it also gives you deep training sets from the knowns in order to predict the unknowns, which allows you to start understanding the 3D structure through predictive analytics that are based on the best training sets in the world, because it comes from that collection. Ultimately, that's a key part to industrial scale microbiome mining, because when you go to that kind of quantity of material, you are increasing the order of magnitude by each doubling of the quantity you receive. So to get to 8 orders of magnitude from the native culture is actually hard, but easy and sensible.
Mm-hmm. So would you say that is the core engineering innovation that allow Bioresortia to move beyond the lab and achieve industrial scale of harvesting? The leap that creates the potential for, or as you just said, the 8 orders of magnitude improvement?
It does. So we like to talk about 3 pillars at Biosortia. You've got the technology industrial scale microbiome mining that brings you the molecules and brings you the data. That's key. The second pillar is unparalleled data depth, training sets, and the data itself that allows you to then use the third pillar, which is computational in silico de-risking. Ultimately, now you can predict structures at a higher probability because you've got brilliant training sets that came from the technology. And being able to predict the structures allows you to start predicting things like ADME, toxicity, targeting, protein binding. You can now computationally de-risk molecules themselves. So having these 3 pillars lined up, each of them are dependent upon the pillar in front of it. You just can't have great predictive capability unless you have the molecules that came from this rich, deep, dark data of the microbiome.
You've mentioned about the data as the pillar. So about AI and data, you said that you're doing complete AI data sets. How exactly does Biosourcer leverage AI to analyze the massive volume of novel small molecules you're obtaining? Is AI used more for target identification, lead optimization, or managing the sheer complexity of the data?
All of the above. So, because we have got data sets that are unparalleled, we can curate data sets for better prediction. So, we can use external data sets, but finely curated, let's say positioned for what would be natural. We don't want to be using a made up molecule from a synthetic data set when we're trying to analyze the molecules that exist in nature and what they do. So ultimately, we can go as far as anybody with richer data when it comes to in silico de-risking and taking that all the way to predictive capability and prioritizing the molecules. High priorities, unmet needs are our focus and our interest, or what our customers ask us to potentially solve for them. So our goal is not to become a pharma company, but it's to become the expert in industrial scale microbiome mining and pulling these molecules out, de-risking these molecules, derivatizing these molecules, and hitting the targets. So the computational stack, the in silico stack, includes derivatization so that we can make sure we can coordinate the brilliance of nature to work best within how it would work within a human.
I'm just going to switch to the business of scale. You've successfully secured a 20 million funding. What is the biggest strategic challenge in translating this technological leap into a successful commercial model? Is it partner identification, IP protection, or managing the licensing process?
Well, with all things in life sciences, we have a community that has learned to do what has been taught before them. So I don't want to call it necessarily bias, but the reality of it is hard in life sciences to bring a new technology to bear in a community that is established. Give you an example. Antibiotics took 40 years before it started saving lives. When you look at genomics, it was poo-pooed by major organizations across the world until it wasn't, and it was accepted. When you look at mRNA vaccines, for 10 years, that was voodoo science that was never going to be material. The reality of it is to do something new in a deeply embedded, entrenched system where funding is based on peer review and nobody knows what the new technology is. So funding new technologies in life sciences is not like funding new technologies in technology, electronic technology, or technology. It's much harder. So our 20 million went to establishing and building the core capability of industrial scale microbiome mining, proving it, and then basically we're now on the path of directly trying to find customers, pharma companies that are trying to expand their pipeline and can seek our ability and our knowledge to help them find molecules that hit their targets. We also have found molecules of interest that we have put into opportunities that we would share with someone who looks to expand their pipeline. So we have found what we believe is an ideal proof of concept in a weight loss molecule series that is not a hard-hitting GLP-1, the orthosteric site, but it hits the allosteric site. But at the same time, this single molecule coordinates multiple coordinating around loss, so that it really is like a knit glove approach, the way nature designs molecules, polypharmacology of a single molecule to multiple targets to achieve ultimately the weight loss. So provide an answer to hunger, to satiety, to hedonic drive, to energy expansion, to happiness, and to potentially, if I continue to eat too much, I might feel ill. So instead of a single target, single molecule, we found one in nature that actually coordinates the way nature designs its molecules, multiple targets within that endocrine system.
In terms of challenging the statu quo for their strategic vision and future impact, you state that it's time for life sciences thought leaders to accurately name the problem in discovery, calling it linear scale issue. What is the mindset shift you are to drive in the industry, and what are the issues for companies that continue to rely on older, less scalable methods?
So I would say one of the biggest issues is the right data in the right place. So I once had of one of the top 4 major pharmaceutical companies say to me, we've got all the molecules we could possibly want. And I was trying to explain that you must have the right molecules. Having molecules is easy in this synthetic world. In fact, you could have more molecules than there are atoms in the universe. The reality of nature's finite, our explorations today are in the infinite. We need to ground the infinite with the finite in a hybridized solution. So it's not about having molecules. It's about having the right molecules and the right knowledge around molecules to advance the systems dramatically. And that's really what we talk about is nature designed and built these molecules, and we need to know more about that in order to build a hybridized process of leveraging those molecules synthetically. So I'm a big believer in using the technologies that exist today, but using them in a focused manner to achieve specific results built on the knowledge that we should have and need to have.
Thank you. You said that you're looking for partner search. Is it what type of collaborator for early licensing and royalties that you're looking for?
So in our belief, it's come from the entire ecosystem, but it's going to come from those that are willing. So when we look at collaborations with universities and research centers, if they have a need, they can reach out to us and we can discuss it. If we look at pharma companies, they have a need, they want to fill their pipeline or support their pipeline or expand their pipeline in a particular area, they could reach out to us and say, here's what we're looking at. Can you help us? We can give people a go, no go predict on our current data sets and our current knowledge. So just recently, we have been able to do some of that at this early stage on areas of high priority unmet needs. That we think is one way to move forward. We do recognize that we're not an academic institution or a nonprofit, meaning our data sets are not going to go into the public domain. We kind of look like what we're doing with our proprietary capabilities as being a service provider to the industry. Anybody can come to us and ask if we can help them. We'll see if we can find a way to help them. If we can, we'll determine a path forward. And that could either be paid up fronts or that could be collaborations and ownerships. There's lots of ways to solve this problem and work with everyone. We want to be able to do that.
Based on your experience and building multiple successful ventures, what is the single most important piece of advice you would give to an entrepreneur or scientist trying to commercialize a truly disruptive, high-risk, high-reward technology?
That is multifaceted. Have bullet points there, but let's try to get it down to 2. I would say tenacity, determination as one bullet point. You must recognize you're going to run into barriers you never believed would be there. So you better be determined. The second thing that I would say is have a real focus on a sustainable business model. Understand that if you're trying to become a business, it's very different than being in the academic or research world. You must be able to create a product, a service, or a technology that people want and will pay for. That's critical.
Thank you. Well, thank you so much for sharing your vision today. And where can our audience learn more about Biosorcia?
Yep, just going to biosorcia.com and hit contact. We'll follow that up with a meeting.
Thank you so much. Nice talking to you. Have a great day.
You too. Thank you.